Good morning, everyone. To begin our conference, we welcome Dr. Jocelyn Smith, who will be updating us on the evolving situation with European corn borer. Dr. Smith is an assistant professor in field crop entomology in the School of Environmental Sciences at the University of Guelph, Ridgetown campus. Dr. Smith's research focuses on management strategies for insect pests of field crops, corn, soybean, and cereals, and resistance management for insect-protected transgenic corn. There's a timer there. Okay.
Yep.
Give you 5 minutes.
Yep. Okay, thank you. Good morning, everybody. Greetings from Ontario. I was here, I think, 6 years ago now to talk to you mainly about Western bean cutworm back then, and seed treatments, the neonics, which was a hot topic then, and a little bit about a new discovery that was happening where we found Bt-resistant European corn borers in Nova Scotia for the first time. And so today I'm going to give you an update on this whole situation and what's been going on with that project for the last 5 or 6 years. So I want to thank you all for the invitation to come back and talk about this. Here's the flashback stuff, some old pictures from 2018 when I went to these fields in Truro. Was kind of shocked to see a lot of corn borer injury in Cry1F or Herculex plants. This was, I mean, when I started in this business like 20-ish years ago, I started scouting corn borer as a summer student for OMAFA, and then it kind of disappeared for most of my career in Ontario, but coming out here and seeing this was quite a shock to that much injury in the field. So just to give you a little background so we understand all the terms that I'm going to talk about in my talk, we need to know the Bt traits involved, and I know this can be kind of confusing at times for growers, but the story starts back in the early 1990s, right, when the Monsantos and Eric's going to correct me, he knows better than I do about some of the companies back then, but they came out with this transgenic corn, the first protein that they could take genes and put into the corn plants so that the corn produces these proteins that are toxic, very specific to certain insects, and the primary target pest for all Bt corn from the beginning was European corn borer. This historically has been one of our worst corn pests in North America. It's been in North America for over 100 years, and it was always really hard to control because of its life cycle, which we'll talk about a little bit later. But Bt corn was this revolution because now the insect feeds on it. It's going to produce these little proteins that bind to the insect's gut. The lining of their gut basically causes little pores to form inside there, and then their guts leak out, and things leak back in, and they die from sepsis, essentially. So that's how it works. Cry1AB was the first protein that was commercialized by Monsanto and Northrop King back in the day in 1996, and it was sold in hybrids that were called like yield guard corn borer originally. Dow AgriSciences, a few years later, commercialized their own cry protein that they was called Cry1F, and that was sold in hybrids that we called Herculex. And then the newest proteins that target European corn borer are from Monsanto as well, Cry1A.105 and Cry2AB2. Cry2AB2 has been kind of the wimpier of the ones, so they're always stacked together in this transgenic event. Cry1A.105 kind of does the heavy lifting, and that's what you find in your double pro hybrids up till this date. Okay, so that's the background. We're going to come back and talk about those proteins again. You know, this Bt corn story has been super successful in North America and a little bit in other parts of the world as well, because all of those proteins, those 4 proteins I just mentioned, we call them high dose against European corn borer, in that corn borers are super susceptible to them, and they will kill more than 99.99% of the corn borers that eat these proteins. So that's why it's worked so well. No other Bt proteins for any other corn pest have ever worked as well as these ones. The rootworm proteins don't work this well. The proteins for corn earworm, western bean cutworm, don't quite work this well. So this was the home run from the beginning with BT corn. And we moved from selling those individual traits in corn hybrids to pyramiding them, so you have more than one in the same plant, right? And that helps, gives you more modes of action. The chances of resistance developing are much lower. And it also allowed us to drop that refuge. If you remember way back when you had to plant a structured refuge with these single toxin hybrids, you were supposed to have a 20% refuge. When we started pyramiding them, we said, well, we can probably drop it down to 5 because the chances of resistance happening are very low. And we can go to a refuge in the bag strategy, which was nicer for everybody, right? The other thing is, since the beginning of BT corn, part of the requirement for authorization of the products is that we have, the governments, CFIA in Canada and the EPA in the States, require that we monitor for resistance in the pest population all along. And nobody found any cases of resistance with European corn borer to these proteins from the beginning. So it was super successful, and we got to a point in time, and we're there now for the last 10 years or so, where about 85% of the corn in Canada and the US all produces these BT proteins that target corn borer. Okay, but so now we find the first case of resistance here in Nova Scotia, which was kind of surprising. And the thing that was also quite surprising to us was that we learned that there were still being sold and planted a lot of corn hybrids out in these short season markets that just only produced one protein at a time still. So there were some really popular Cry1F hybrids that were being planted here. And that could be part of the reason why this happened, because in every other major corn growing part of North America, we had shifted to the pyramided products. So our resistance risk was going down, but yours, unfortunately, you weren't getting those options out here. You were only getting primarily these single toxin hybrids, and we think that's part of the problem. So once this was discovered, they said, okay, we got to pull those off the market right away, and they replaced them. Really the major one that they had in the pipeline to replace a single Cry1F hybrid with was a Cry1F, Cry1AB pyramid. Okay, so that happened. Now, the other thing that happened was we started a lot of new research because, we thought we had corn borer pretty well licked for a long time, but now it's kind of making a comeback. So we have collaborated with a lot of folks in this room, and we've started to do a lot more research on the pest, and we're going to talk about that now. I want to show this picture because here are a lot of the folks who have been working with me on this for the last 5 years or so. Caitlin and Jason here from Nova Scotia and New Brunswick, Suki in Prince Edward Island, some folks in Quebec, Julien Seguet and Brigitte Duval, Gilles Canal, he's a retired OMAFA consultant near Ottawa in Winchester, and Holly Biker at Winchester, our station. Tracy Bowdy is the entomologist with OMAFA in Southern Ontario, and then out west, John Gavlovsky in Manitoba. So all of these folks have been helping us kind of restart all the corn borer research in the last few years. Our first research objective I'm going to talk about is that we wanted to now increase our resistance monitoring again. We had been doing it all along, since 1996, but we were focusing our efforts back then on where's the most corn, thinking that if resistance is going to happen, you'd think it would happen where most of the corn is, where most of the BT corn is being used. Like you'd think it would happen in Iowa or Southern Ontario before it would happen in Nova Scotia, right? Anyway, so we really needed to expand our scope of resistance monitoring, and we do that in a couple ways. One is that we get field collections from different areas across the country. We bring them to my lab in Ridgetown. We rear these cages full of corn borers for a couple generations, and then we do these diet bioassays with them where, let's see, does this work? Yeah, on these trays. We put diet in the tray, overlay the protein on top, put one little corn borer in every cell, and then after 7 days, see how many die and weigh them and all that stuff. So we can get an LC50 value, right? What concentration of protein kills the population, kills 50% of the population. In this example, these data are from the original detections, so you can see two lines on the the left side of your screen, those are susceptible populations from Ontario, where we exposed them to Cry1F. You can see increasing concentrations of Cry1F across the bottom there. And those populations are all dead at a really low dose, right? Our LC50 for them is about 6.5 nanograms per centimeter squared of this protein. But these lines across the bottom are the we had 4 collections from Truro and 1 from Kentville from 2018 that were like flatline, no response to Cry1F. They're totally resistant to it. So we want to calculate a resistance ratio with these populations, where we take the LC50 of the field population, divide that by the LC50 of the lab colony, the susceptible ones, and anything over 10 we would consider resistant out in the field. And these Nova Scotia populations were well over 30 times more resistant back in 2018. So we've been doing a lot of these tests, and now I'm going to show you a bunch of tables that show our resistance ratio values for all 4 of those different proteins I mentioned earlier: Cry1F, Cry1AB, 180-105, and 2AB2. Sorry. So here you can see, and we have 2 controls we're comparing them to. That's what the C1 and C2 mean across the top. Anything that's orange here has a resistance ratio greater than 10. So we think they're resistant out in the field, we're going to have problems. If they're a lighter colored orange and a little asterisk beside the number, the resistance ratio is not greater than 10, but it's significantly higher than the susceptible population. So this is kind of like early warning, maybe something's happening here with these populations already. So you can see back, these are 2018, those original 5 collections from Nova Scotia, very resistant to Cry1F, and a couple little things that look funny on Cry1AB and Cry1A.105. We weren't able to get all the proteins done for every population every year because rearing insects is challenging and so on, but this is where we're at. So 2019, we do our expanded collections. We got a few from Quebec. We've got a lot more from Nova Scotia. We have a couple from New Brunswick, one from PEI, and one from Manitoba. And the highlight here, so again, we see some resistance in Nova Scotia mainly. And you notice here, we see a lot more increasing tolerance to Cry1A.105 with those Nova Scotia populations. And then it was interesting that we've actually got a population resistant to Cry1F from Quebec this year as well. This was near Montreal. And they're also somewhat tolerant to 1A.105. Okay, so there on the map, as you can kind of see our original resistance detection over there in Truro, and then this new one over here near Montreal. We don't know if this is spreading or not yet. 2020, we didn't have a whole lot of success in collecting, but what was interesting was a collection that came from Manitoba, not even from corn. John had trouble finding any corn borers for me that year, so he got some out of millet, and they looked actually resistant to Cry1F. So that was kind of a surprise. And that, there's no way the corn borers traveled from here to there, so we think this is probably a separate problem. We come back to Nova Scotia in 2022, and we went to the original kind of area in Truro where we found the original resistance. And you know, I mentioned earlier that we've removed the Cry1F hybrids, and we've replaced them with 1F, 1AB hybrids, right? So we go into one of those fields, we find quite a bit of injury in the fields, like more than just your 5% refuge in a bag plants. So we sampled a population out of that field, and that's the one down here where we found to be very resistant to Cry1AB as well. And that's the first time we've ever found that. It's also the only time we've ever found it. We haven't found it again since, but it's a bit of a warning, okay? 2023, you know, I think at this point, we're just seeing that things are increasing in New Brunswick as well as Nova Scotia. And then 2024, kind of the same story. And I guess the interesting thing from 2024 was that we finally got some populations from Alberta, from a collaborator near Provost, who were growing a lot of, non-BT corn, but just starting to grow some BT corn out there, and they were seeing tons of corn borers in their fields. So they sent us some, and they looked very susceptible to everything so far, which is good news. Now that was in this area here, near Provost. But in 2025, we now have a couple collections from southern Alberta and one over here in Saskatchewan that we're testing right now. Preliminary data, but I've got concerns. It looks like we might have some problems happening out there now too. So, there's a lot of whys. Why is this happening? But we could have a kind of a common theme in these shorter season markets where the single toxin hybrids were planted for a longer period of time. Okay, so we're doing this lab testing of all these populations. We're also doing sentinel plots where we plant strips of BT and non-BT corn out in the field so that we can get field relevant exposure, because those protein concentrations we use in the lab could be not exactly what the plants produce. The plants could be producing less. So we want to see what happens out in the field as well. There was a program started a few years ago from a professor at the University of Maryland. He designed this to actually look for resistance in corn earworm originally, but we adapted it for corn borer. And you can see on the map here the sites where we've had sentinel plots since about 2020 now. Ridgetown and Winchester in Ontario, one in Quebec, and then Jason runs one usually in Sussex the last couple of years. Caitlin has been doing some in Nova Scotia, and Suki did one for the first time in 2025 in PEI. Anyway, these sentinel plots are interesting because we get a lot of variability in corn borer populations. To be honest, we get nothing in our Ontario plots. There's just no corn borers still in Ontario, really. But we come out here and we're shocked to see the amount of corn borer pressure that you've got. The other thing I want to point out here is that in 2023, a collaborator put out one of these plots in Connecticut for the first time, and she saw corn borer damage to BT hybrids there. So on Cry1AB, and Cry1A.105 and 2AB2 hybrids. So that's the first detection in the US at all of this BT resistance. And maybe there's some connection with the East Coast here. We're not too sure yet. But here's some data just from the last few years, the sentinel plots in Kentville. The corn borer pressure has been kind of low. I know 2025 was a tough year. Those plants weren't all that attractive because they were so dry and small, right? But Jason's found a hot spot in Sussex where we see quite a bit of injury, like we measure the amount of tunneling within each plant. We have to, break the plants down and measure these corn borer tunnels. And you can see quite a bit of injury here to Cry1F plants, which is this yellow bar. The purple is Cry1AB, and the blue is Cry1A.105 and 2AB2. So, industry sometimes wants to kind of fight over the definition of how much of this is bad out in the field. And in my mind, we just don't see any of this in most parts of corn growing regions. So to get this much injury is definitely significant, and something's happening. So that's kind of the summary on the resistance at this point. Just to make this quick, I would say that we've seen Cry1F resistance now in Nova Scotia since 2018. In Quebec, we found it in 2019. We've seen it again in 2024. We're still going through our 2025 collection, so I don't have all those data yet. We found some Cry1F resistance in Manitoba in 2020 and New Brunswick in 2024. When I say resistance with the red letters, that's meaning that's what we're seeing in the laboratory bioassays. When I say we see unexpected injury out in the field, that's more of what we're seeing in the sentinel plots. So we're definitely seeing a lot of Cry1F problems in Nova Scotia and New Brunswick. For Cry1AB, we've really measured resistance from a population in Nova Scotia in 2022, and we do see unexpected injury in New Brunswick, Nova Scotia, and Connecticut. And then for 1A.105 and 2AB2, we see this resistance in Nova Scotia and New Brunswick. We see some feeding on plants in New Brunswick, Connecticut. And even though we see feeding on the 2AB2 plants, we've never been able to find resistance in the lab to 2AB2 yet in Canada. However, now that we've got the attention of all the US corn entomologists too who work on this, there's a collaborator in Minnesota who went out and started looking, and he's found 2AB2 resistant populations in Minnesota and Wisconsin, which is kind of interesting when we have not seen that here in Canada. So I think kind of the one thing to note here is okay, there's actually a lot happening with corn borer resistance. It's early days, but it's building. And I think we just weren't looking hard enough for the last number of years. I mean, it's inevitable that eventually insects always develop resistance to everything we throw at them, right? But you guys were the red flag, so I'm sorry to say, you kind of set off a whole nother wave of corn borer resistance monitoring. So the next objective to our research is to figure out more about the biology of the corn borers that you have out here in the Atlantic provinces. We used to know a lot. We thought we knew a lot about corn borer biology, but most of that was figured out by entomologists in Iowa in the 90s or before, right? And so things are different out here. It's a totally different landscape. And corn borer is a really complex, interesting insect. There's different pheromone races, like some of them are attracted to certain pheromone blends over others. Some of them like just corn, some of them will feed on like 200 different host plants, including, I'm sure we all know potatoes, right? Peppers, tomatoes, hemp, millet, all kinds of things. So we're trying to learn more about the corn borer biology. The pheromone trapping was kind of an old way that you would monitor for corn borers. That's not turned out to be that successful in recent years. So the commercially available pheromone lures don't seem to work all that well anymore. So we're trying to figure out a better lure. We're trying to figure out which trap corn borers prefer to fly to. We've got a couple examples here. These both work fairly well. And so we've also collaborated with my friend Kelsey Fisher there in Connecticut. She's making new lures for us to test that we're using out here. And this data are pretty interesting, where she's got a bunch of collaborators in the US as well as out here in Canada. And look at where all the captures are of corn borers. I mean, we were the only place that found any in the last couple of years. So that tells us something. The corn borer pressure out here is totally different. Yeah, so we'll learn more about these lures hopefully in the next couple years. From these pheromone trap data, we've been trying to figure out, what is the biology of the corn borer out here. It looks like primarily it's probably just one generation per year. And this little schematic kind of gives you the life cycle breakdown. So they overwinter as a late stage larva. They're about 1 inch long in the corn stalks. They tend to go down low in the plant, like the bottom 30 centimeters or so, to overwinter in the corn stubble. And then in the spring, they'll pupate in there. They fly out of the field, and they go look for mates outside the field. They don't stay in the field. They go to grassy ditches and stuff. That's where they like to find, they call them action sites for corn borers. That's where the action happens. And then they go to find a field that they think is going to be nice to lay their eggs in. So they do that in probably July or so, and then you see their eggs hatch out into these tiny little caterpillars that burrow into the plants, and that's where they are for the rest of the season. And they do tunnel up and down, as I said. So that's why you get all these problems with stalk rots getting into the plants, lodging, stalk breakage, ear drop, more mycotoxins in the grain because they have these little exit holes where the fungi can get in there. So, and of course, yield loss is part of the package here too. All right, and then lastly, we're trying to characterize the resistant populations. This is more of the kind of the real sciencey stuff, not so much, that the farmers care about, but we want to know how does this resistance come about, and what does it mean? What implications are there for resistance to the other traits, et cetera. So I had a grad student, Emily Glasgow. She took some of the resistant corn borers from Nova Scotia and made crosses with a susceptible population from Ontario so we could look at the inheritance of the trait and also the fitness of these resistant populations. So this is kind of preliminary as well, but she looked at how many eggs do they lay, how many days are they laying eggs and things, and she found that the ones that have resistant mothers, the resistant females, Cry1F resistant, I should state, just to be clear, they produce like twice as many eggs as the susceptible females do, so that's not good. They live a little bit longer than our susceptible strains, and they emerge a little bit later, so you probably are getting kind of an assortative mating situation where the Cry1F resistant ones are going to mate more with the Cry1F resistant ones than the susceptible ones. And these things all kind of indicate that, oh great, these traits are going to spread faster in the population, right? A postdoc in my lab, Dr. Yasmin Farhan, she also did some back-cross experiments to look at how this Cry1F resistance gene is inherited. She's determined that it is just a single gene problem. It's autosomal, it's not sex-linked gene, and it's recessive, which is kind of what we expect with a lot of these. That was what we predicted would be the case. Then she compared the genome for a receptor in the gut of the corn borers to see if there were differences in the resistant ones to the susceptible ones. And she found 6 mutations in the gene here for this ABCC2 gene, it's called. And one of them, the one I highlighted in red there, results in a protein truncation in that receptor. So the receptor is somehow getting modified so the Cry1F can't bind to it to the same degree as it should. So in the resistant populations from Nova Scotia, we're seeing a really high frequency of that mutation, and we don't see it we see it to less of a degree in the Quebec population, and we don't see it out in Manitoba. So this is kind of like telling us that the resistance that developed out here is probably different than the resistance that's developed out west, and maybe there is some connection between the Quebec population and the East Coast. Okay, so that's lots of stuff to take in about what this BT resistance means. What does it mean for you as growers, right? Well, we're concerned that this is going to bring back the corn borers to some degree across North America. Of course, that increases our risk of yield loss, mycotoxins. Maybe we're going to go back to trying to spray for these things, which does not work all that well, because they get inside the plant so quickly. Timing insecticide application is hard. Like I say, they're not just a corn pest. This could increase risks to other commodities as well. And if organic producers want to use dipel or some BT foliar insecticides, this resistance will also probably confer resistance to those organic pesticides. So those are some problems. Here's another one that I want to point out as corn producers. So here's our current list of the hybrid pyramids that we have. Right, we have these Cry1F, Cry1AB hybrids, which are what you find in any Acre Max products from Pioneer, AgriSure products from Syngenta or NK. We've got the Double Pro in the center, which is our 1A.105 and 2AB2, and then any Smart Stacks Pro, PowerCore, Vorseed, all the really stacked up stuff, they all have Cry1A.105, 2AB2, and 1F in them, okay? So if now we have resistance to Cry1F, we've lost that trait in this pyramid. So really we're putting a lot of selection pressure just on Cry1AB now, and we're starting to see some of that tolerance tick up in some of the populations we're collecting from the East Coast. We're going to lose it over here too, right? So then you're relying just on the 1A.105 and 2AB2. So that's not great. But then even worse is that we're seeing this trend that with Cry1F resistance comes along cross resistance to Cry1A.105, right? And so now we're going to lose this one. So that really doesn't leave us with a whole lot in our toolbox to control corn borer going forward. Like I said, 2AB2 is not the best of all these proteins, so leaving it on its own isn't ideal. And as far as we know, there are no new traits coming for corn borer in the next 8 to 10 years at least. And what we do know is coming might not be quite as good as these original ones were, but who cares? At least there's going to be stuff coming eventually, but it's a ways away. So what can you do? At this point, we're asking you to look for corn borer injury, or have somebody scouting your fields and have a look for it. Hopefully I've shown you some good pictures today so you know what to look for. And if you find it, the seed companies want to know. They do want to know where it's being found, and they might give you some recommendations about what we can do around management, but we are talking with growers about maybe controlling corn borer the old-fashioned way, which could involve chopping corn stubble, right, with flail mowers or some other kind of tillage implements that would kill a lot of them when they're overwintering. There's a program that Bayer has started in the last couple of years, 2023, with AgCall, a company from out west. They've hired a bunch of people to scout fields. They started doing this in 2023 in the East Coast, and it was really successful. Now they're expanding it all the way to Alberta. So they've got AgCall scouts across the country, and their aim was to target like 10% of their acres, because they're really concerned about losing Cry1A.105. So if you get a call from somebody who says, can we come scout your field for this program, it'd be really great if you could participate in that. And the last thing I'll say is that, we're kind of ending this first phase of all the corn borer research and hoping to go into another one. We want to continue the resistance monitoring work. We want to delve deeper into the corn borer biology in Atlantic Canada first. And I'm planning to connect with a couple researchers at Dalhousie, Dr. Chris Cutler, who's an entomologist there, and Yunfei Zhang, she is here somewhere, corn agronomist, crop physiologist. We're going to work together with some students that we'll have at Dalhousie to do some more in-depth corn borer research starting this summer. We want to look at what is the yield loss associated with corn borers nowadays in our modern corn hybrids. Maybe it's not quite as bad as it used to be, because our corn hybrids are a lot better than they used to be. And then look at how we can kill them when they're overwintering, and more research to do on these corn borers that have multiple Bt resistance, not just Cry1F. So I want to just lastly thank all of the folks who have helped so much with this project. There's a huge list there, as you can see, but the Atlantic Grain Council has been a big support, as well as the Nova Scotia, New Brunswick, and PEI folks. So thank you very much. I don't know, do we do questions? Oh, we do, okay. We have time for questions. Nope. Okay, yep, thanks.